Hello everybody. My name is Nilufar Monhasari, and in the following approximately 40 minutes, I'm going to present you our comprehensive solutions which help you to improve your clinical grade regulatory T cells Treg manufacturing. I would like to start with the important disclaimer that states Miltenyi Biotec products and services are for research use only and not for therapeutic or diagnostic use, unless specifically indicated. Please acknowledge that. Thank you so much. Before going through the scientific discussion and introducing our workflow and products and so on, let's begin with a brief company introduction. At Miltenyi Biotec, our mission is to advance scientific research and medicine by providing solutions for curative cell and gene therapy, as well as biomedical research. Miltenyi Biotec was founded at 1989 by Stefan Miltenyi, is the largest owner-managed company in Germany, and we have two main sites in Germany. Bergisch Gladbach is the headquarters. Tetero is the GMP manufacturing facility. And we have subsidiaries in 28 countries and distributing our products to 73 additional countries. And we are over 3,000 employees all over the world. At Miltenyi Biotec, as you can see in this slide, we cover different topics and support our customers with translational portfolios and workflow. Okay, let's have a look at our agenda for today. First of all, I would like to give you an introduction about regulatory T cells, then talking about clinical application of Tregs. I have three main topics for today: graft-versus-host disease, rejection of solid organ transplant, and autoimmune diseases. Afterward, I will go through our clinical Tregs manufacturing workflows, and at the end, I would like to sum up with the most important points and closing remarks. Introduction to regulatory T cells. Tregs are important subset of adaptive immune cells and control immune reactions for maintaining homeostasis in our body, and they have three main functions. The first one is suppression of, you can see here, the suppression of overwhelming immune responses which keep the balance and homeostasis in the body. The second one is suppression of immune responses against self-antigens or non-self antigens, which protect against autoimmunity and mediate the tolerance. The third main function is suppression of anti-cancer immune responses, and it supports the tumor growth and is not the favorite functionality of Tregs and needed to be considered and tightly controlled during the cancer treatment. As you can see here in this picture, all the T cells develop in thymus out of a common precursor cells, and depending on the stimuli, they can develop to the CD4 or CD8 T cells. Treg classified according to the development origin into three subtypes: T Tregs or N Tregs. They are thymus-derived Tregs and known as naive Tregs, and are the majority and most stable population of suppressor Tregs. P Tregs, they are the P Tregs, they are the T cells that's in vivo, preferably induced to Treg, and they may convert to effector cells afterward. I Tregs are induced Tregs that you can see here. They are the T effector cells which can be induced in vitro in a specific condition like the presence of a special cytokines and so on to Tregs and called I Tregs. But how can we characterize Tregs here? As you might know, there is no unique surface marker for Tregs. They are characterized by the combination of surface markers. In general, Treg cells express CD4 and CD25 and identified by the absence of marker CD127, and the naive Tregs can be identified by the additional marker CD45RA. Moreover, FOXP3 is an intracellular marker of Treg and expression of that used as a proof of functional Tregs. Tregs has crucial roles in different types of diseases and provide novel therapeutic strategies for treatment of different immune disorders such as inflammatory disease, tumor immunity, transplantation, autoimmune disease, and so on. According to key roles of Tregs, it can be studied from different aspects and in the context of wide variety of diseases. So my question to you is that from which point of view are you studying or using Tregs in clinic? The good news is that Miltenyi Biotec is covering all aspects of a Treg study from preclinical to clinical. Here I would like to use this great publication in Cell that was published some months ago, which discussed about Treg and CAR Tregs as the effective and novel immune cell therapy methods regarding to autoimmune diseases and also graft-versus-host disease and organ transplantation. I'm going to show you an example in more example, let's say in details in the following slides. This picture here shows the healthy situation, that there is an immune balance between regulatory T cells and T effector cells. However, in some circumstances, the balance is disturbed, like GvHD, autoimmune disease, or organ transplantation or rejection. And the reason which underlies the immune imbalance are diverse. And adaptor immune therapy with Treg cells lead to the reestablishment of immune balance, and it's the reason that why Tregs are very attractive candidate for cellular therapy. Let's start with some example of clinical application of Tregs. First topic is GvHD. What is GvHD and how Treg therapy can help in this case? Hematopoietic stem cell transplantation, HSCT, is an established procedure concerning the infusion of autologous, syngeneic, or allogenic stem cells for several high-risk hematologic malignancies. The success of allogenic HSCT depends on a multitude of parameters: type and stage of the underlying disease, age of the patients, human leukocyte antigen HLA disparity between host and donor, and intensity of the pre-transplantation conditioning regime. This main side effect of HSCT is graft-versus-host disease, GvHD, where donor T cells recognize the host minor and major histocompatibility antigens and proliferates, damaging target tissues. Patients undergoing GvHD receive an immunosuppressive regime, which is called steroidal therapy. Steroids are a kind of medicine called an immunosuppressant. This medicine weakens the new immune system, so your new cells don't attack your body. Starting GvHD treatment as early as possible can lead to better results. But studies showed that the post-transplant outcomes, depending on the initial disease status, for example, only 50 to 80% of patients with acute GvHD and 40 to 50% of patients with chronic GvHD respond to steroidal therapy. So there is a need for alternative and more effective strategies to modulate the ongoing immune response. Use of regulatory T cells as a cellular therapy for treatment of GvHD is one of the efficient and known alternative therapy for limiting immune responses for prevention and treatment of GvHD. As you can see here, for production of clinical Tregs, physicians take the blood from the donor and isolate Tregs, and then with or without expansion, infuse the allogenic Tregs to the patients for prevention and also treatment of GvHD. As you can see here, there are three main points regarding to Treg manufacturing which should be considered: the purity of the cells, cell number, and specificity. For GvHD application, we need to have intermediate to high purity to mitigate GvHD symptoms, avoid transmission of EBV or other infection to patients. The second factor is the cell number, which should be high to increase the probability of target tissue interface. And the third one is that it's better to have polyclonal Tregs in this application. In this way, more alloantigen-specific Tregs can be targeted. So for GvHD, mainly scientists, physicians use the entire Treg and isolate the whole Tregs out of the PBMCs or blood. The next example of Treg therapy as an immune cell therapy is related to the rejection of solid organ transplantation. The main reason underlying for the rejection of an organ is often an inflammation of the transplanted organ. After organ transplantation, the immune suppressive co-treatment can either successfully protect the organ or the organ get inflamed because the endogenous immune cell recognize the new organ as foreigner and attack it. And it happens in 17% of the patient in the first 3 years post-transplantation. In this case, blood collected from this patient and Tregs are manufactured for autologous blood products, and several publications showed that this therapy is possible during and after the time that the patient treated with immune suppressive drugs. And Treg can transfer to suppress the ongoing inflammation. And in this area, there are several ongoing studies, clinical studies, for the application of Tregs and also CAR Tregs as a novel immune cell therapy. Let's see what are the criteria for Treg manufacturing in the case of solid organ transplantation use. Purity needed to be high or ultra pure to avoid co-enrichment and expansion of inflammatory cells. Cell number needed to be very high to increase the probability of target tissue interface. And we need to target the antigen-specific Tregs in order to direct Tregs to find the right organ. And the last but not the least topic is the application of Treg therapy for autoimmune disease. Autoimmune diseases are when the own immune cells, T cells mainly, recognize own tissue as foreigner and attack it. In the recent years, several teams with very nice data and publications showed the success of Treg therapy regarding to the immune diseases such as diabetes type 1. And you can see here, blood needed to be collected from patients, then Treg are isolated, and then autologous Treg will be injected to the patients to suppress that highly activated patient's immune cells. And what are the key criteria for this application? It's more or less similar to organ transplantation situation. Purity needed to be high or ultra pure to avoid co-enrichment and expansion of inflammatory cells. Cell number needed to be very high to increase the probability of target tissue interface, and we need to target the antigen-specific Tregs in order to direct Treg to find the right organ. Many of you are already in the field of clinical Tregs manufacturing and application and know already about the challenges in the fields. As you can see here, I listed the main challenges like contamination, inconvenient isolation procedure, maintenance of Treg function after expansion, and also maintenance of Treg phenotype after expansion, isolation of Treg subsets like naive Tregs, which is a very small fraction of Tregs, ultra-pure Treg population. Okay, but how Miltenyi Biotec can support you to achieve your goals and also overcome the challenges? We support you with highest technology and best quality products, which I'm going to introduce to you in the rest of my presentation, such as the CliniMACS system, our reagents medium for expansion of your cells, our great new cell sorter, which is very well known in the Treg field. Let's start. I would like to start with this slide just to remind you that we have a translational workflow and support your study from research to clinic. And today, as in the title of my talk, we focus just on the clinical part. But if you are interested to know about our research products, portfolios, you can visit our webpage, and also my recent webinar is available on YouTube and on our webpage as well. You can have a look and get more information on that. Okay, here you can see all the products that you need for clinical grade Treg manufacturing in a single frame. We support you all the devices and reagents that you need for Treg isolation, Treg activation expansion, and last but not the least, Treg analysis. And here you can see the two main workflows that we offer you according to your application and needs. But the main point that makes these two workflows different is related to the isolation step. As many of you might know, Treg is a rare population of the T cells in whole PBMCs, and isolation of them is really challenging. At the same time, they are really fragile and sensitive cells. So, workflow 1 and 2 answer your needs. Let me just explain you in the following slides. Workflow 1, isolation based on CliniMACS Plus, which is a dominant and very well-known device in the field regarding to the Treg isolation, and you can find it in most of the clinical Tregs application. CliniMACS Plus enable you to first deplete CD8 and CD19 cells, and afterward enrich your CD25 positive cells. As you can see here, it is mainly used for production of cells in the field of GvHD mainly. And also you had an intermediate to high purity, and approximately the purity is reached up to 96%, and the whole process takes about 7 hours. As I've showed you in the previous slides, we offer you CliniMACS reagents for the isolation like CD8, CD19, CD25, and you can see how it works in the flow cytometric plots. By getting the high CD25 expressing cells, we can target Tregs, and after enrichment, we reach the purity of approximately 92%, which is a great number for Tregs. Okay, regarding to CliniMACS Plus CD25 system, let me summarize what the system offers you. Convenient isolation process, high cell number, the phenotype of your cells is maintained as well as the functionality, and you can isolate polyclonal Tregs. And you can find online the reference list, which is a very nice list of publications that you can see which teams, how they use this process. But there are some challenges in the clinical grade Tregs isolation, which are not really solved and touched by CliniMACS Plus CD25. But the good news is that we have an alternative for you, which gives you more support. The main challenges are listed here. Isolation of naive Tregs is still a big challenge in the field. They are a small cell population, and maximum recovery and purity is needed. Ultra-pure isolation of Treg is stressful for cells, and also it is very time-consuming. The field needs to have GMP flow sorter to get the approval of clinical studies faster and easier. Altogether, to solve these challenges, we offer you the workflow 2, which contain two main devices, CliniMACS Prodigy and MACSQuant Tyto cell sorter. These two devices help you to first isolate Treg subpopulation and antigen-specific Tregs, and also enable you to produce ultra-pure Tregs. Workflow 2, isolation based on CliniMACS Prodigy LP25 process and Tyto cell sorter. Using CliniMACS Prodigy LP25 process enables you firstly enrich your CD25 positive cells, secondly labeling your cells with fluorescence color, and thirdly elute the cells in the special Tyto buffer. And altogether, your final yield is ready to be run in the cell sorter to have ultra-pure antigen-specific Tregs. As you can see in the chart, the main application of the workflow is related to cases that scientists and physicians need antigen-specific ultra-pure Tregs, more than 98% purity, which is a promising number. And the whole process takes about 10 hours. Again, in a single frame, you can see all the reagents, buffer, tubing set, and whatever you need for isolation of your Tregs with this workflow. I would like to just give you some introduction about CliniMACS Prodigy for some of you that doesn't know about it. The CliniMACS Prodigy offer a flexible platform to cell processing application, enabling the magnetic separation of different cell types as well as customized cell processing protocol. As you can see here, Prodigy provides you a sterile, closed, portable, flexible, reproducible, with high safety and traceability system. And the specific process for Treg enrichment in Prodigy calls LP25 process. In the following slide, I'm going to explain more about this process. Okay, as it is summarized here, the starting material is mainly leukapheresis, the total volume, the total cell number, volume, and everything is specified here. I just mentioned to that you will receive this presentation after my talk, and you can really go to the details and have a deeper look on the numbers. But as you can see, the final product is amazing. You will get 60 million Treg cells and 20 million naive Tregs approximately. The process time is about 4 hours, 1 hour hands-on, and 4 hours automated process. And in this a little bit complicated picture, you can see how different reagents and buffers and your starting material get connected to Prodigy. Please, no worries, everything done in the first step, and it is not complicated at all as it looks. You will be trained by our application support team, and you will learn it very easily and quickly. No worries. Let's start to have a look at the whole process, but this time step by step. The first step is CD25 enrichment. Before enrichment, 3% of the whole blood cells are Tregs, and after the enrichment, we reach 30% of the whole cells as Tregs. And you can see here very well that naive Tregs are very small fraction of the whole blood cells and ultra pure isolation of them. Now you can imagine that how challenging can be. Next step is fluorescence labeling of enriched cells. We provide you MACS GMP quality antibodies. For this step, please don't forget that all these steps are automated and no hands-on. Just in the first step, you need to connect all the antibodies, reagents, buffer, and so on to the system, and then everything can be done automatically. And the last part of LP25 process is the elution of your target cells in Tyto buffer. Okay, we go to the next step, which is the sorting of the cells. And here I would like to talk about MACSQuant Tyto cell sorters. Many of you are familiar with that, and maybe some of you are not still familiar with our unique cell sorter. I just explain you what makes this cell sorter very unique. In general, it has 3 laser and 8 fluorescent channel, and give you this opportunity to sort your sensitive Tregs in a very gentle way. It gives no stress and pressure to your cells, and according to the microchip-based sorting process, it is completely gentle to your cells. it is enclosed and has disposable cartridge, you just need to transfer your cells to the cartridge and then put it in the machine device and then run the specific program and leave the cell sorter. As I have no time to go more into the detail of this device, I would like to invite you to just visit our webpage and also contact us for more details and use the advantages of this cell sorter, which is really like the best choice for sorting of Tregs, which are really fragile and sensitive cells. For sorting of Tregs and having ultra-pure final yield, I would like to suggest you to use the two-step sorting strategy. First, run the cells for debulk sorting, and then follow that with purity sorting. After debulk sorting, you reach 83% purity, and after purity sorting, your final yield will be pure up to 98%. In this slide, you can see the whole process with exact numbers, like an example for you. That starting material here is 1/4 of Locopac, and it contains 2.5 billion cells. After Prodigy LP25 process, we reach about 38% enrichment, and total Tregs number are 7.7 to 10 power 7. And after cell sorting using Tyto, you reach about 98% Tregs and 2.6 million naive Tregs, as you can see here. And each step takes like 5 hours, but keep in mind that for this sorting, now we have the highest speed cartridge, and it makes the whole process faster for you. Then please visit our webpage and ask for more info from local Miltenyi team. And here, I just wanted to compare these two isolation methods regarding to process time, hands-on, the starting material, and purity. And the main point is that both of the systems are working perfectly, but the choosing of each of them is dependent on your application, depending on your needs and your clinical plan. And for getting more information, to getting our consulting, and if you need that we help you to choose the best platform for your studies, feel free to contact us. We will help you, and we can go through to your studies and your plan and type of application of your Tregs and so on, and you will receive a good comprehensive support from us. Okay. Let's go one step further. Treg expansion. As many of you know, expansion of Treg is another challenging area. However, we provide you with proper medium, cytokines, activation reagents, and so on to facilitate these steps. For efficient culturing and expansion of your isolated Tregs, you need to consider some main factors. The first one is proper cell culture medium. The second one is the cytokine. As you know, the correct dosage, stability, and activity of cytokines are super important to have good expansion of your target cells. And Treg activation reagents, which we offer you MACS GMP ExpAct and also T-cell transact. And Rapamycin is another important factor. Let's go through each of them in more details. MACS GMP medium contains pharmaceutical-grade human serum albumin, optimized formulation with or without phenol red, complete medium with glucose and a stable glutamine, and you need to just choose the proper one according to your application. Quality control functionality test on each batch, which I find it very exciting and great. And filled in flexible bags, which makes it easier to be connected to CliniMACS Plus and Prodigy, and we offer you the customized formats on request. Three other components are MACS GMP ExpAct Tregs, which is the activation reagents for your Tregs, and it maintains FOXP3 expression, and it activates your Tregs to have a very good rate of expansion. The second one is GMP Rapamycin to inhibit the expansion of conventional T cells and to enhance the proportion of expanded Treg FOXP3 expressing cells in culture. And MACS GMP IL-2 that you can see here, the specific activity in comparison to other commercial products, which functionality is really good. Here you can see the post-sorted fraction of Tregs with 98% CD25 positive, CD127 negative, and also here 92% of CD25 and FOXP3 positive Tregs. And after expansion for 14 days in the presence of TexMACS GMP medium, MACS GMP ExpAct Tregs, Rapamycin, and MACS GMP IL-2, we reached 98.3% CD25 FOXP3 positive cells, which is a great yield according to the expansion rate, purity, and functionality of your Tregs. But I would like to mention a point here that please keep in your mind that expansion of Tregs is really donor dependent. According to your donor, that it is a healthy person or patients, and it is even different between healthy people and patients, the percentage of the Tregs in the whole blood cells. And also depending on your manual protocol, there is no right now standard or very close, complete closed system right now. And each team, if you go through the publication, you can see it easily that each team has their own manual protocol. And also, it depends on the duration of the expansion. Some of the scientists think that, okay, when they expand it for a longer time, they will reach a better rate of cells or a better rate of expansions. But it's not true. Sometimes your cells are exhausted. And what I really want to suggest you, before going through the expansion stage, check the functionality and phenotypic of your cells, if your cells are healthy. After some type of cell sorting, in which the cells, your Tregs, suffer from a high pressure of sorting, they are not really in a good condition, and then you don't get a good expansion results. So please keep in your mind all these factors and then go through the expansion. And there is a MACS and more. It's a customer report from Professor Lombardi lab that is in our webpage that you can have a look. And the last but not the least step is the analysis of Tregs during the cell manufacturing process. And at the end of the process, to control the quality of your cells during the experiments. The analysis of Treg can be done in each step of your workflow to check the efficiency of cell isolation and expansion, as well as phenotypic and functionality characterization of your cells. As you can see here, there are sort of markers, and according to your experiment and Treg cell subpopulation, you can design a panel and characterize your cells. But don't forget that we support you to design the best panel according to your application and needs. Feel free to contact us. What we offer you regarding to your Treg analysis. Let me show you the various MACSQuant analyzer, which are fully automated flow cytometer that even include a sorting column as well. In addition to MACSQuant analyzer, that we have different types, different models, and then each one offers you different possibilities. We have the Treg detection kits, which are the pre-titrated antibodies, and also our reaffinity recombinant antibody plus bio-dye antibody conjugation, which are highly pure and has lot-to-lot consistency antibody. For getting more information, again, I would like to invite you to visit our webpage, and for panel designing, contacting us directly. And here I would like to refer you to the poster that I put the link on that at the end of the presentation, which suggests you different panels for Treg phenotyping, and at the same time you can see the gating strategy and the way that we analyze the data and get very good overview on that. And here I would like to show you the functional analysis of Tregs that we recommend you to use the Treg suppression inspector kit. In this kit, we have Treg suppression inspector, polyclonal stimulus, MACSibits coupled to alpha CD3, CD28, and CD2 to stimulate your responder T cells in an optimized way. And this proliferation can be suppressed by functional Tregs. It works like this. When your responder T cells receive this stimulation, they start to proliferate. But when your responder T cells are co-cultured with Tregs after receiving the stimulation, Tregs, let's say functional Tregs, suppress the proliferation of T responder cells. And in this way, you can monitor if your Tregs are functional or not. In this graph, you can see that better. That high proliferation of T responder cells in the absence of Treg, whereas co-culturing of Tregs with T responder cells lead to the inhibition of T cells proliferation. And at the end, I would like to say that this polyclonal stimulus is optimized for T responder cells proliferation and not Tregs. So it's a common question that people ask, okay, when we just stimulate T cells, we stimulate the proliferation of Tregs as well. But in this case, it's just optimized for proliferation of T responder cells. Okay, we are reaching the last slides. Let's summarize here that Miltenyi Biotec offers you a wide variety of products according to your Treg application, and we have solutions for all your needs and pain points. And if you need more information, I put some link here for you, and in the case of any more support, please contact your local Miltenyi team, and also sending us email, calling us. We will be happy to support your studies. And yes, we are done. Thank you so much for your attention.